Good morning, everyone, and welcome to the special talk today. Okay, so today it's going to be like the special talk from Dr. Felix Moronta. But before we go to the talk, so I would like to invite the Vice Director of เกษตรศาสตร์ University Research and Development Institute, Associate Professor วิษณุ บุญญาวิวัฒน์ to give an introduction from Biosafety Committee and Kurdi. Okay, please.
Dear Dr. Felix Moronta, Biosafety Committee, Faculty of Science, and our guests, it is a pleasure to extend a warm welcome to everyone here and also audience who watch our Facebook Live for the special talk titled Cultivating Progress, ICGEB, part in Environmental Biosafety. We appreciate your time, Dr. Felix Moronta, a senior program specialist in the Regulatory Science Group at the International Center for the Genetic Engineering and Biotechnology, or ICGEB. Yes. For being our special speaker today and talk to Faculty of Science for invited us to be a co-host today. เกษตรศาสตร์ University Research and Development Institute, or CURI, support and encourage our เกษตรศาสตร์ University faculty and student researching in many fields of study, such as science, agriculture, science, biotechnology, engineering, and also social science. Not only the provide of fund, but also giving an advice, supervision on research standard. As I mentioned before, CURI have a unit called Research Standard Division and Laboratory Animal Center that on the responsibility in research standard includes biosafety. Now, in time, emphasis of biosafety as a research standard, the research should comply with. So it is relatively new to many researchers to make understand research standard division and laboratory animal center as biosafety regulation office regarding and control biotechnology activity. However, cooperation with Biosafety Committee to encourage เกษตรศาสตร์ University Research to allow for following biosafety rule and regulation and address the key to perform a related research. ICGEB regulation science group activity might be a guidance to improve a biosafety framework in เกษตรศาสตร์ University, building a partnership along with the standard research work in biotechnology and agricultural science field in biosafety, culture, and sustainable development. I hope that we can learn a lot from this event today and be able to create and enhance national capacity in the regulation of biotechnology. Thank you.
Okay, thank you, Associate Professor วิษณุ for the introduction about the biosafety of เกษตรศาสตร์ University. Okay, next I would like to invite Professor Arintip Thamchai-Pinet from Faculty of Science, เกษตรศาสตร์ University to give a welcome speech to Dr. Felix.
Dear Dr. Felix Moronta, our speaker from Biosafety Committee, เกษตรศาสตร์ Institute for Research and Development, and our guest. It is our pleasure and my pleasure, very pleasure as well, to extend a warm welcome everyone here to the Faculty of Science, เกษตรศาสตร์ University for the special talk on topic Cultivating Progress ICGEB Path in Environmental Biosafety by Dr. Felix, a senior program specialist in the Laboratory Science Group at the International Center for Genetic Engineering and Biotechnology in Italy. We are very honored that he has taken his time from your Thai schedule in Thailand to accept our invitation to speak at our faculty here. The Faculty of Science, เกษตรศาสตร์ University has committed in highest standard of research and teaching for all branches of science, particularly biological science and biotechnology, has held a particular place at our university as our mission focuses on cultivating knowledge for agriculture and livelihoods of the country. Biosafety is one of the key areas that our researchers will have to address in their works, and we are at the early stage of development of the regulatory framework for biosafety here in Thailand. We would like to thank Thailand Science Research and Innovation, Thai Young Scientist Academy, and KERDI for help organizing this event. We, the Faculty of Science, are happy to host everyone here and hope we can learn a lot from the event today. Thank you.
In brief, with the next session, I would like to invite Associate Professor Visanu and Professor Arintip Thamchai-Pinet to give the souvenir to Dr. Felix in this for the special seminar. Okay. Dr. Moronta, just a little bit, okay, in brief, okay, before you give the talk. Okay, Dr. Felix Moronta is a biologist committed to facilitating the adoption of agricultural products enhanced by modern biotechnologies. He received his PhD from University of Alicante, Spain, and now he works as a senior program specialist in the Regulatory Science Group at the International Center for Genetic Engineering and Biotechnology, ICGEB, in Trieste, Italy. He works closely with national and international authorities to ensure regulatory effectiveness of biotechnologies. He also served as a co-chair of the Global Young Academy, which is the organization that mobilizes and empowers young researchers from 6 continents to lead international, interdisciplinary, and intergenerational dialogue. Okay, so everybody is ready for the talk? Okay, so I would like to invite Dr. Moronta to give your special talk. Thank you.
Thank you so much. Thank you so much for the invitation, for your kind introduction, for your kind words. That was an amazing welcome. From now on, I would like for you all to be relaxed, because I would like that this talk is very close between you and myself. I would like that this is an exchange of knowledge. I'm looking forward for learning from you about your backgrounds, your expectations, your knowledge, what's going on here in Thailand, because I'm not aware of your situation. This is normal, and so on. And because of that, because I would like this to be a close exchange, I will ask you, the people that is more far, if you can come closer, because that would be better, please. Like a couple of seats at least. And also because I would ask you several questions, and I would like to write your feedback there. Yeah, thank you. And yeah, before starting with my presentation, I would like also to bring you a personal context. You have heard about some of my professional achievement and so on, but what have brought me here is my personal history. I am originally from Venezuela in South America. There, my profession is biologist. I studied biology there, and then, well, I have been scaling in the academic career and so on, doing my PhD in Spain and then my postdoc in Italy and so on. But the thing that changed my life is when I was involved in agricultural studies in my country, in Venezuela. I went to the rice paddy fields in my country, and I went there with the mud until my knees to sample rice plant, because I was interested in the microbiota that was associated to those rice plants. And this work, when I meet closely the needs from the farmers, the gaps in the rural part of my country, and so on, was the thing that makes me realize that I would like to work beyond the scientific research. So after many years, etc., I was able to leave the research lab and dedicate myself to the science policy arena. So I'm currently work in the, let's say, in the middle way between science and policymakers, international policymakers. I'm not in, my institute is located in Italy, but we are an international organization. So I work with our member states, because we are an intergovernmental organization, but also with international agencies, mainly from the United Nations. So when I am in this high profile fora, etc., I always try to remember my days there in the rural part of my country and what brought me there. With this introduction, I will start my presentation. Just an introduction of the work that in my group we are doing around the world. So this is our path in environmental biosafety. So, agriculture. Agriculture is not a luxury. We need to eat, and we need to produce every day more food in a sustainable way, ideally. However, the agriculture has several problems associated as any human activities. When I said problems, I am thinking in environmental problems. One of them could be the land use. The agriculture is one of the main drivers of the biodiversity loss, and mainly because of the changes in the land use. We need to cut trees to produce more food. It's not a luxury, we need to eat. The population is growing, and so on. And not only the land use, but also the use of fertilizers, the use of pesticides, because we need to protect our food. Because, yeah, insects, fungi, etc., also likes what we eat, and we need to protect them. For we to be able to have food in our tables at night for our family, they need to be protected from insects. And also the use of water. They use a lot of water, fresh water, and it's perhaps easy to think that the source of water is every day more scarce. So, with this scenario of many problems, and we need to really take care of what we are cultivating there, is where agriculture is developed. And there are alternatives, there are solutions, there are many ways to try to overcome or to alleviate these kind of problems. One of these alternatives, one of these solutions could be the biotechnology. I don't know if you are more or less aware of how biotechnology could help for food production. And I would like to hear from you your thoughts. There are not wrong answers, absolutely not. I just want to hear from you if you think that the agricultural biotechnology can offer solution to those problems. How and which kind of approaches? Come on, give me some idea from the audience. Any thoughts? Okay, I can help you. Yeah. Yeah, perfect. Yeah, that's a good alternative, how biotechnology can alleviate that. Using several tools, absolutely. Okay, and actually where I wanted to go is to present you with true cases, true real case scenarios of biotechnology products that are addressing these kind of problems. Here, this is cotton in Colombia, and this cotton is a genetically modified cotton that is resistant to pests. So this cotton doesn't need so many insecticides. It needs less application of insecticides. And this is the same for the maize. This picture was also taken in Colombia, and this maize is called a BT maize because it is resistant to a pest, an insect of maize. So this maize needs less application of insecticide. In this case, this is papaya in Hawaii, and this papaya is resistant to viruses. And again, the viruses of plants are transmitted with insects, so a normal control for this kind of diseases is insecticides, and this papaya doesn't need insecticides for that diseases. And this is in Bangladesh, eggplant in Bangladesh. Again, this is a culture that is resistant to pest. These are biotechnology products that are being cultivated and produced and commercialized in some countries in the world. And these are examples of, again, genetically modified crops or GMOs that has been produced in developing countries, that has been commercialized by smallholder farmers. We are not talking about big companies. We are not talking about monopolies. We are not talking about anything like that, like in the beginning of the 90s. We are 30 years ago of the big controversial aspect of the GMOs. We have evolved that part, I think. Actually, there are data now, after almost 30 years of commercialization of genetically modified crops, that suggest some environmental benefit of the cultivation of this kind of crops. Here, there are some just examples of one of the last reviews of the data, of the historical data on the reduction of pesticide use, increase in yields, increase in farmer incomes. And again, we are talking about developing countries mainly. And actually how some authors are suggesting of how these biotech products are supporting the Sustainable Development Goals from the UN. Okay, this is some of the benefits, but of course this is a human technology and there are risks associated, and there could be a bad part of these products. So, thinking about genetically modified organisms, GMOs, genetic modification in crops, foods, again, I would like to hear from you what are the potential risks that you can think of using, of cultivating, of eating, whatever, these kind of crops. Please, let me know something from you. Potential risk. There is no wrong answer. Let's brainstorm a little bit. Okay, change in soil biodiversity. Yeah, something else? Other risk? Okay. Okay, yeah, yeah. Okay, I will summarize the 5 main risks, and these 2 are included there, not the soil and wild populations. Gene flow. Gene flow is one of the main concerns, that your genetically modified crop can, we are talking about plants, can pollinize a wild species, or wild not, a non-modified species. Another concern is the horizontal transfer, and this is, again, the genetic modification from the plants going to bacteria, for instance, the soil microbiome, because bacteria can incorporate nucleic acid from the environment. Another risk, it is the wilderness or invasiveness of your genetically modified plant. What if goes out of control and then reproduce as crazy? decrease biodiversity, because we can talk about monoculture on this kind of culture monopolizing the entire ecosystem and so on. So this is a true concern, a true risk. And, well, non-target organism, that your genetically modified plant could negatively affect other organisms. In the case of the crops resistant to insects, just thinking an insect that is not a pest also could die from eating that plant. No? This is one of the risks that could happen. Okay, now talking about risk. What is risk? No? So a risk is a process, risk and the risk assessment. A risk assessment is a process used to identify potential hazard and analyze what could happen if a disaster or hazard occur. And this is an important distinction, extremely important distinction. It's not the same a hazard and a risk. A hazard, you know, there are sharks in the ocean, but they are there and we are here. So the risk is almost zero. The risk becomes a reality when you enter a water that is infested of shark. So the risk is the probability for a harm or hazard to occur. It's the exposure. The risk is about the exposure. And this is something that we are going to talk in a little bit during the talk. During the talk, because it would be, I have focused the talk on risk assessment of this kind of biotech products. And general points of the risk assessment. Any request for an agricultural biotech application, I mean this is for cultivating a GM crop that is going to be used in an unmanaged environment. This is to be released in the environment. This should be evaluated within a risk-based regulatory decision-making process. And most of the country, including Thailand, has in place this kind of decision-making process, or how to evaluate. And the decision-making will be influenced also by ethical, sociocultural, epidemiological, ecological, and economic consideration. This is a multidisciplinary evaluation. And this decision-making should include mechanisms that facilitate the effective engagement with all the stakeholders involved and integrate all these considerations in the overall decision-making processes. And the risk assessment is based in the best available scientific evidence. And this is aimed at informing decision-making processes. And the risk assessment methodologies and outcomes may differ between countries and their regulatory authorities. So it is important for you and for me to understand that in every country where I provide this kind of talk is different, because every jurisdiction has their different rules and regulations and laws. There are some international agreements that I will comment it later, but in essence, every jurisdiction has their own rules. So the key questions of the risk assessment, there are 4 questions. What could go wrong? Just imagine again the eggplant that is being cultivated in Bangladesh. What could go wrong with that cultivation? So an identified harm occurs due to the exposure or interaction with a modified organism. How likely is the harm to occur? It's not enough that could go wrong, that everything could go wrong, but how likely is that? So an identified harm is assessed with regard to the likelihood of the occurrence of a series of individual causal steps in a pathway that may lead to harm. A pathway that may lead to harm. The assessment will derive the chance of harm from the overall series of individual steps. And then, if the harm occurs, how serious is that harm? The seriousness of potential harm can be assessed via an evaluation of the consequences should the harm be realized. And then the level of concern. The level of risk is evaluated as negligible, low, moderate, or high, by considering a combination of seriousness of harm in the likelihood of its occurrence. So, let's think in a hypothetical scenario. Again, the eggplant in Bangladesh is resistant to an insect because it's toxic for a specific insect. Let's say, I can imagine, okay, I don't want the ants to be affected by this crop. No, because ants, as every animal, are important in the ecosystems, but a non-target organism, the ants. Just imagine in the hypothetical scenario that the ants are susceptible to these toxins. And, well, it results that one ant is killed because this toxin. How serious is that one ant is killed? Instead, how serious is an entire population of ants is affected? So the seriousness is very important. And the risk assessment is always comparative. We need to compare to weigh what is happening with the conventional counterpart of the genetically modified crop. What is going with the conventional, currently cultured eggplant with a genetically modified eggplant. We cannot compare our GM crop with an unrelated species, with an unrelated crop, with a very far away variety of eggplant. We need to compare with the maternal material of that eggplant. So the criteria for harm and likelihood are used to establish a baseline for a comparative risk assessment. Using the parent organism, including a standard management practices and the receiving environment. And this helps to specify the type of change due to biotechnology that would be considered significant in terms of potential harm from the modified organism. Example, conventional crops already present risk to the health of people. We can talk about the gluten in wheat, or allergens in soy, in peanuts. The peanuts are mortal for some people. But still, there are no problem of being cultivated and commercialized. Or to the environment, the weedy characteristics of some plants. So our conventional varieties, some of them are really, really harmful for some people. So, but does the modified crop possess an increased risk or an additional risk to the already those that are present? This is the kind of question that always the regulators in national agencies need to think about it. So the risk analysis is a complex process, and it is summarized here, and it is loaded with unfamiliar terminology for us scientists, but also for the regulators that are there in the ministries in our country, And the stage of the risk analysis, well, it is the key issue identification, then the risk assessment, risk decision, and the risk communication. I'm focusing in the first square here, key issue identification. You both have told me about potential risk, but it is not enough. It is not enough now. It's the correct way to think in risk is thinking in protection goals, and the protection goals are dictated by the laws in our country, in the Thai constitution, in the Thai laws about environment. There, you need to find the protection goals. Something that is quite universal is biodiversity protection, environmental protection. Every country has in their constitution this kind of protection goal. So what are management goals? And what are assessment endpoints? The identification of assessment endpoints, it is the most difficult but the most crucial component of risk analysis. The soil biodiversity, how is it measured? No? What is the quantification of a soil diversity that you need to keep there? How you can measure the genetic flow in the how you measure that? You need quantification. You need an assessment endpoint, and this is crucial. And as I said, it is the most difficult, but it needs to be there because again, the risk assessment is a science-based process. And how to make sense of this formative step in the risk assessment? Well, there are one approach that is called problem formulation. I will tell you now, and because of that I have brought the flip chart here. Okay, a problem formulation. First, you need to identify your policy objective, again, that is dictated by the national legislation, usually. And these two are quite common in many constitutions, strategic plans, national strategic plan, national laws, etc. Then, based in this objective, you create a list of undesirable harms. I don't know, for instance, in the countries that have, I can think about Mexico, where the maize was obtained, no, 1,000 years ago. They are very jealous of their national maize seeds, and they don't want to any contamination. And also they have the monarch butterflies, and they want to protect them. So you create a list of the things that the things here and the things here that you for sure want to protect. And then the undesirable harms. And then you reformulate the harm as a catalog of hypotheses, the crazy ideas. Hypotheses are crazy ideas. So you can think, okay, the eggplant that is cultivated here will be eaten by this whatever insect that I like so much, and the total population of this insect will decrease, and this will have deleterious consequence for the environment. This is a statement and hypothesis. And then each hypothesis is confronted in a pathway to harm. Okay, I am signaling here a pathway to harm because I want to give you a couple of examples of how the risk assessors in the ministers of your country go through this process. Testing the pathway to harm. So again, identify key steps in the pathway and add available data where pertinent, determine if the existing data break the pathway, and if not, identify what additional data would allow testing of the hypothesis. Okay, then in a more simple way, let's think something unrelated. A highway, outside a highway. What could be a harm of having a highway just next door the university? So we can think that, okay, a student could cross and be killed by a car. So it's a true harm that is there. We don't want for this to occur. So a pathway to harm would look something like this. So the hypothesis will be that the highway outside the campus yeah, this is a very unfortunate hypothesis, but this is the kind of things that regulators should think about. What is the first step for this harm to occur? What should happen? For this, the very last step, I don't know the number, would be, well, a student that unfortunately is dead by a car. So, but from this to this, there are several steps that need to happen. Let's say, okay, and a student goes out. No, this is the first, in all the serious steps. And then this person needs to cross the highway but in a prohibited zone, no? So it's not just a crossing, it's cross the highway, I don't know, wrongly. But then you need cars there, of course. Cars, high speed, of course. High speed. And then you need both at the same time. We were talking about interaction. We were talking about exactly. Let's say an encounter. The speed, the attention of the driver, and there are many sequential steps for a harm to occur, many, and you can think about it. Here could be sub-steps, and so on. And then when you already confectionate your pathway to harm, you need to assess the likelihood of each step to occur. How likelihood is this to occur? This one, this one, this one, this one, and so on. If the result of all this, is that the likelihood of this is very high, are you going to prohibit the construction of the highway? Or no, you only implement risk management methods. You put lights, you put announcement, you put a crossing over the highway. So if the risk to the harm is high, then you need to apply risk management methods. But I did okay. And this is something that regulators also do with genetically modified crops. This is a true case in one of our workshops on risk assessment. The hypothesis was that cultivation of a drought-tolerant maize leads to loss of genetic diversity of the land races. So then you can see the pathway to harm. The drought-tolerant maize is planted in the same area as the conventional maize. Then it is needed that the farmers prefer growing this genetically modified maize instead of the conventional one. And then with time, farmers will stop growing certain land races, I mean the classical maize varieties. And then farmers do not store or multiply their land races to maintain viability. And all these sequential steps will result in the loss of land races and hence genetic diversity. Again, this is a true case of a pathway to harm. And then what is important? Okay, we have developed our pathway to harm here. I don't want to, I'm not going to repeat it. And then the level to harm. Okay, so harm could be, oh, this is, sorry. Okay, it's cutting. Okay, just 1 minute because there are one important row there that is not showed. Why? Okay, now you can see the 4. Yeah, okay, marginal, minor, intermediate, or major, and the description of each of those points. And it is split between health and environment. For instance, I can see here in our case, of the unfortunate case of the highway and the student, the level of harm will be major because it implies the death or the ill, an ill that is irreversible. And you can, in the case of health, human health, you can see the comparison between marginal and major harm. And the same for the environment. A major harm in the environment is when there is an extensive damage to the whole ecosystem, communities, or entire species that persist over time. So in the case of the risk of the gene flow or the soil biodiversity, it would be a major risk. I mean, are we talking about an irreversible negative change in the entire ecosystem? And so on. So with this very simple concept, it is possible to identify the level of harm. So you have determined the level of harm. In this case, it would be major. But then it's not enough. Then you need the likelihood for those to occur. And here you have the description of the highly unlikely, unlikely, likely, and highly likely harm. In this case, well, let's say that here it was major and highly likely. This is just an hypothetical situation. And there are also, there is the possibility of a quantitative instead of qualitative likelihood. And here, some regulators propose to to make an assignation of percentage. But what if a risk falls at 79%? Is that likely or very likely? And what if a risk, is difficult for percentage? And the recommendation is to continue to work with qualitative assessment. And once you have the level of harm and you have the likelihood, then you can use the risk matrix and definition. In our case, we determine that the harm, if occurs, has a major effect, and if it is highly likely, then the level of risk is high. Here, talking about the highway. Here, with this example, we determine between major and highly likely that the level of risk is high. In this case, well, it is good here. Depending on the level of risk, there are a series of recommendations. In our case, the risk is of considerable concern that is unacceptable. Again, unless action of risk management are applied. Coming back to this example, then this was determined by the regulators. This would be very likely. This maize, because it has a benefit for farmers because it needs less water. It's highly likely for this step to occur, and then it was not possible to determine the likelihood here because they needed more data, because each of the steps should be confronted with data, scientific available facts. They determined that this would be likely, this unlikely, and the final step highly unlikely. And considering both the minor risk and the likelihood, they determine that the risk is negligible. So the risk is not discernible concern, and there is no present need to invoke action for mitigations. So, and the risk assessors in this case recommend, this was the recommendation for the decision makers. Okay, and all this topic of risk assessment is being dictated, or this approach of risk assessment is being dictated by international agreements. The Cartagena Protocol on Biosafety establishes a very basic procedure for performing the risk assessment. This is not enough information, and because of that, countries request, for instance, our help in my group to understand better how to perform risk assessment. And Thailandia is a country signature of the Cartagena Protocol. And this was in the past, 20 years ago. This is an application for a company that wanted to cultivate GM crops, presenting the application to the national regulatory offices. This is the amount of information that just one plant variety needs to be assessed. The amount of information today is even bigger, but it's not in paper anymore, hopefully. So far, questions? Because I have finished the topic of risk assessment. Any question from the audience on this topic? From the online audience, something? Okay, so I can change the topic because I want to focus the last minute of my talk. And what is regulatory science? This is the name of my group. Regulatory science, there is a generalized definition by the FDAs of the United States, and is that the regulatory science is a scientific discipline consisting of the development and application of scientific methods, tools, approaches, and other relevant processes derived from various scientific disciplines used to support regulatory and other policy objectives. An abbreviated definition is that regulatory science consists in the application of science to support policy, notable regulatory objectives. And these objectives provide a basis for improved capacity for safety and efficacy evaluation and monitoring of candidates and licensed products. This is general, agricultural, pharmaceuticals, whatever. Modernize current regulatory pathways or frameworks, develop new regulatory pathways where there are no currently. So the role of my group is assist our member states, we are an intergovernmental organization, to identify, regulate, manage, and monitor biotech products within their own jurisdictions. And I have brought some examples of our activities. Development of regulation, assisting the development of effective safety and regulatory system for products of modern biotechnology, like biosafety legislation and administrative system, communication strategies and tools, training, both academic and vocational. For instance, the regulatory experience exchanges. We organize exchanges between regulatory office, between countries, usually between a country that has a mature functioning regulatory offices with one that is being developed. And the objective is to improve the capacity for safety and efficacy, evaluation and monitoring of candidate, again, unlicensed product. And well, I already told you about this objective. Also, institutional capacity enhancement. We provide, for instance, guidelines for the institute be strengthened. It doesn't matter if in a ministry, if the personnel change with the government, it is important for the institution to be strengthened. We also have developed an e-learning portfolio on biosafety, and with this module on risk assessment, confine and containment, GMO food safety, biosafety and biosecurity, environmental risk assessment, socioeconomic consideration, and biosafety regulatory framework. And this is a e-learning portfolio that was developed with for our African associates, but now we are happy to extend the users of this e-learning portfolio. We have developed an additional module on public health and epidemiology because of the pandemic. And this is an example of one of the last capacities enhancement activity that we have developed. Well, literally, we went well, not around the globe, but in several places to provide training on risk assessment. This is 1 week training on risk assessment. And well, we went to Senegal, Nigeria, South Africa, Turkey, and in Philippines 1 year ago, well, a little bit more than 1 year ago, to provide this risk assessment training to governmental officials, to regulatory offices. Also, we collaborate with the Convention on Biological Diversity to develop this comprehensive report on synthetic biology. Synthetic biology is an emerging discipline that is so much in the focus now in international agencies because of the uncertainty on how to regulate this kind of products. So we produced this comprehensive report for them, and it's being used by diplomats and international policymakers to get information on SynBio. And also, again, with the Convention on Biological Diversity, we have developed guidelines for the risk assessment of this crazy technology that is called gene drives. In this case, animals are being genetically modified to transmit their genetic modification to the entire offspring. This is something amazing. I mean, crazy and amazing. I don't know, because it's a huge scientific achievement. This is a non-Mendelian, no? From Gregor Mendel, non-Mendelian mechanism of transmitting the genetic information. So, theoretically, with the release of one single genetically modified gene drive organism, you can, by some X number of generations, have an entire population that harbor your genetic modification. And this technology is being developed for trying to reduce the incidence of malaria in Africa, for instance, with a public health approach. And not only for biodiversity reasons, for fighting against invasive species, for instance. We also recently developed a communication strategy for the Biosafety Clearinghouse. This is a platform for exchange of information between countries on biotechnology, from laws and the scientific part also. And we also worked with countries, and this is just one example. We worked with Panama. We were with them for 2 years, updating their biosafety regulatory framework and providing them with training and manuals and procedures on how to assess not only the genetically modified crops, conventional, but also the new ones that are coming. I'm talking about gene editing, CRISPR crops, SynBio products, etc., to be future-proof The regulation, most of the regulations that are in place now are from 20 years ago, from the very conventional genetic modification, and the technology and science evolved so much and so fast, faster than regulations. And this is an example very similar to the country one, to the Panama, but this was with a regional focus. We were working, again, like 2 or 3 years with the Caribbean countries for developing a common understanding on biosafety regulatory frameworks. And this is a current project. This is the International Union for Conservation of Nature. These guys are an international organization, extremely important organization, because they are the one that assess the world biodiversity. And they are the ones that are informing the UN on the biodiversity status in the world. And because of the importance of the synthetic biology, we are working together, IUCN and my group, we are helping them to develop policies in synthetic biology. And because of this project, I am in Thailand now. Next week, we will have a training in the Asia regional offices of IUCN that is here in Bangkok. And here we were in Nairobi with the Citizen Assembly of the IUCN after having the training in Simbayo. And in the next week, I will be with the same group, but in the historical city, I don't remember the name, in the north of Bangkok, the UNESCO site. Yes, exactly. So I will repeat this kind of picture, but after the training, of course. Yeah. Okay, more questions about activities of my group. Okay, we are about to finish. And this is my institute. No, we are in a small group offices in my institute, the International Center for Genetic Engineering and Biotechnology. Is an international organization somehow associated with the UN system. We have so far more than 80 signatory states, 80 and 60 member states, and 3 components here in Trieste, in Italy, where I work, but also we have a component in Cape Town and also in New Delhi. Thailand here is a signatory country, but we are waiting the ratification for becoming a full member state. And the full member state, okay, this is not updated, but we provide PhD fellowship, postdoctoral fellowship. We provide also research grants to research groups, and also meeting and courses that are over the world. And our member state can apply for any of those programs. And well, the potential areas of collaboration of my group is on GMO detection, scale dependent for nascent GMO regulatory system, but also having a hierarchical approach with regional center of excellences, networking, consultation, and technical help in developing regulation or implementation of regulation in biosafety and also in biosecurity. This is an aspect that I didn't bring here, but we are also working with both branch of the safety, biosafety and biosecurity. And why collaborate with ICGEB? Well, we have a dedicated staff serving to our member states. We are highly regarded by international community in providing high quality scientific and technical training in biosafety, and a strong association with established GMO regulatory agencies all over the world. And well, we complementary and close collaboration with technical assistance providers. So we have a huge network of experts in each of the regions, because we understand the cultural nuances of working in, I don't know, sub-Saharan Africa and the Caribbean, in Southeast Asia. and effective technical and finance administration in ensuring successful implementation of project. This is my group. Currently, we are only 4 people. And well, with all this history, starting from the problem with the agriculture, I hope that our path in environmental safety is a little bit clear that at the beginning of my talk. Thank you so much.
Your valuable talk, and I think for myself I gain more information about like how to make the policy. Okay, so right now I would like to move to the Q&A session. Okay, so if anyone that has question, you are welcome.
Dealing with GM of like, okay, when GM you have transgene and you have to assess the effect of the transgene on the environment, the presence of transgene, right? But what about genome editing? What's the trend in regulation in your perspective?
Everyone is wondering the same. Everyone, even myself. So, yeah,
So please
there are 2 trends now. No? A precautionary trend and a innovation supportive trend, let's say. For instance, Europe has decided that genome editing will be regulated exactly as GMO, regardless of if they have foreign genetic material. It doesn't care. Doesn't matter. They will be GMO. And as Europe, New Zealand has done the same until achieving an agreement, a better understanding. When some uncertainties are more clear, they prefer to put everything in the same sack there. This has not scientific sense so much. But okay, this is their decision, is their jurisdiction, that's fine. Other countries instead has been more pragmatic, saying this has not foreign genetic material, this cannot be differentiated with a conventional breeding method, because currently agronomers, they use chemical, they use radiation for generating plant varieties, and these methods are introducing mutation there, and this genome editing product cannot be differentiated from a conventional breeding of genome editing. So some countries like the US, Japan, Argentina, and some others, there are more countries following this trend, saying no, this is not GMO. They are going to be regulated as the conventional breeding plant material. So we have these 2 trends. But I think that the second one is the one that that is being considered much. In Africa, there are already two countries that decided the same, that they are not genetically modified organisms. Yeah, because they prefer to assess the final product. It doesn't care how do you get it. No? It is the product, not the process. Instead of other countries as Europe, they care so much about the process. How do you get there?
Follow-up question from that, because I think like with the regulation that you have to be able to detect it first, right?
You have to be?
You have to the detection problem. So in
yes.
order to regulate something, you have to be able to detect it if it's something that you need to regulate or not. So I guess like with detection of the genome edited product compared to no, it's compared with the mutagenesis, like product from mutagenesis. I don't know. I still can't think of the way to tell the difference between them in terms of like, in theory, maybe in terms of likelihood, but in practical way. Do you have any feedback regarding that aspect?
Yeah, the detection. Yeah, that's one of the key aspects for the relation. Yeah, you are completely right. The conventional GM crops can be detected using PCR and very simple molecular biology techniques, and that's not the case for genome editing that has not foreign material. In some cases, as Europe, because they have a lot of money, they are suggesting to sequence the entire genome of the organism, because genome sequencing is every day cheaper and etc. They are, yes, or trying to sequence a determined region, but yes, they say yes. Let's try to sequence.
Do sequencing is
yes,
not for
the
us
organism
every day.
to try to catch one nucleotide that has changed there. In other cases, because of the impossibility, it's not practically feasible. No? So this is one of the drivers for some countries saying, okay, no, it's not possible. So this is not a GM. Yeah. Thank you.
Okay. Any more question?
Dr. Felix, I know a lot about your group work activities and also the cultivating part in a lot of developing countries. So I would like you to share about the first step when you go into that developing country, how you push the researchers or the farmer to know the knowledge or to know the regulations that beyond only the scientific area, but between the politics and science field, how you cultivate and address that the keys of biosafety into them when they do a research. Please share us. Thank you.
Yeah, thank you. I like how you say how I push them. I would like to, come on, do it right, but I cannot do that. The member countries call us. They request our help, directly or indirectly. I mean, our member states could go directly to us, requesting this technical help and so on, or what has happened, and perhaps is the more efficient way, the member countries request the help to the UN agency, to the Convention on Biological Diversity, to the UN Environment Program, that are the ones, in high profile, with their tentacles in their member state in the UN, providing technical help, managing, implementing projects for strengthening these regulatory agencies. And then the member state ask the UN, okay, we need help because we don't know how to do this, this is very complicated with that, and the UN call us, the ICGEB, and then we go and please do it right. Yeah? So that's the two way. Yeah. There are many other ways of establishing collaboration, but these two are the ones that have brought these multi-year projects and global perspective projects. I mean, I just brought some examples, but we have other collaboration with research institutions and university, smaller, etc. So we are open to many collaborations. Yeah.
Thank you. So is it possible to be that the gsci could be the lead about the cultivating biosafety part in Thai? Is it possible that if we only the Ksesa University or only the faculties of us need your help, it is possible to send to UN and
Yeah, absolutely. Just in the hypothetical scenario that is not possible, we build the possibility. So the important thing is the establishing the common interest and the relationship and trust building, etc. And then we make it happen. Absolutely.
okay, thank
Yeah.
you. Hope to work with you in the future.
Okay, I have a question, Dr. Moronta. Could you share like your experience when you like try to regulate, or I would say like facilitate to the scientists? Like, is there any like difficulties or burden when you talk to the scientists? I mean, for me, when I have like a scientist hat, I feel like this is too like complicated or a lot of like data or work or documents that I have to submit. So do you have any difficulties to like deal with that, deal with this situation? Yes.
Yeah.
Anyone stubborn with you?
Honestly, I haven't found difficulties. No, because the people with I work with, perhaps they, from the scientist's point of view, they already know how to do the things in the regulatory aspect. So I have not had the opportunity to work with a scientist that is completely unaware of how to comply with the regulations. What I have faced is the other way around with the policymakers. That's a very challenging, because we scientists and policymakers, we don't speak the same languages. So the understanding is extremely difficult.
Are you sure that you don't speak in the same language or the same language, but just like...
Exactly. Yeah, exactly. So in my experience, it has been more difficult with policymakers instead than with scientists.
I see.
Yeah, but this is personal. Perhaps my supervisor, my boss, this is the brain of the group, a brilliant person. Perhaps she could have another experience, other kind of perspective. Yeah, with scientists.
Sorry, another question on different aspects. So it seems like part of your work is about science communication to, well, of course, policymakers, so they're like non-science people. So do you have any tips or tricks in terms of how to communicate with like, how to communicate science with the non-science audience, especially policymaker? Any recommendation, any suggestions?
Yeah, good. Yeah. Get training. This is something that is not good to improvise communication with policymakers. It's extremely important. Go and take a training on science communication for policymakers. Yes. A science communication training, whatever, it is useful, of course, and very frequently they have an aspect of science, of policymakers, but there are other cases where they are focused on policymakers. It's completely different. I have taken training on science communication before transitioning from a lab to the science policy arena, and it was like that, science communication and just one module or whatever on policymaker. But now I have experience. Well, I have a concrete example. I went 3 days to Brussels. It was a science communication for policymakers specifically on genome editing. was very targeted, and it was amazing how to draft the messages, how to approach them, what kind of messages, what kind of language, vocabulary, etc. Yes. And I mean, you take the training, and this doesn't mean that the next day you are the communicator. No, you need to put it in practice, and you will get wrong, and you will do it wrong, but you need to practice, practice, practice. Yeah.
May I ask who are policy makers? I mean, actually, it should be us, which is like scientists in the major fraction, right? I mean, this is from what I think.
Yeah.
Yes, but I know that this happens all the time. Like we will be like covered by someone else to regulate us. So what about you, who are like a policy maker?
I have a contradictory thought on that, because yes, absolutely, scientists, we should be part of the policy making processes. Absolutely. Or ideally, the policy making process should be informed by science and scientific evidence. But several times this is not the case, because politics and everything else is happening. But usually, policymakers, in my case, in my work, are officials in ministries. They are there trying to apply, draft the policies. But then these policies, it doesn't mean that they are going to be approved. They need to go to other processes of developing policies. And well, eventually, the politics will come, and etc., etc. But yeah, the important thing is that science should be part of the policy making processes. And I'm not sure if science should be the only one, because I mean, we need social aspects in the policy making. So we need social scientists, the ethics, socioeconomics, many social aspects that must be there. Yeah.
I'm sorry, I'm curious, one more question. Your organization, the other side, do you have the role to like communicate to the social or to the consumer about GM crops or biotechnology stuff or not, to like at least to give them like a knowledge whether or not it's good or bad to use the product?
Yeah. No, because
do you think we should do that too? I mean, like in terms of like institute or university.
Okay. It depends. It depends of the mandate of the institution. In our case, because we work with governments, some governments are contrary to the technology, but still they need to have regulation in place. We need to protect our neutral stance. So we cannot work in a communication strategy for promoting the technology. This is something that we don't do. Absolutely. We promote and we advocate for the biosafety, how to do it right, the scientific aspect of the technology. But if you need to be sure of the consumption or whatever, that's in an arena that could be perceived as you are advocating for the technology and
I mean, I know like to push like GM crops or biotechnology stuff to the social. So at first, I think that we have to like educate them first.
Oh yes.
Right.
Yeah.
Like how this thing can be like come to your table, and how did the scientists do that?
Exactly.
They let them like make the decision whether they want to eat it or not.
Exactly. And well, one of the examples that I always try to use is that you are crossing that bridge every day, or you are putting your children in the school transport because you trust the driver, you trust the engineering that built the bridge, you trust the pilot of your plane. The same with our regulatory offices. They are experts. They're analyzing the risk assessment data. If a product is in the market, well, we should trust that it's safe.
Okay, all right. Is there any more question? Okay, so I think like that's all for today.
Yeah.
Thank you,
Thank
Dr.
you.
Moronta,
Thank you
for
so
giving
much.
us like very nice talk.
Yeah,
Okay.
thank
As I
you so
said,
much.
like I think I have like some or more idea like in my head for the risk assessment and risk management.
Okay.
Thank you
Yeah,
very
thank
much.
you.
Okay.
Thank you all.
So, งั้นพูดเป็นภาษาไทยลองโอเคค่ะก็คือสรุปทางไหนยืนตรงโพเดียมหรอ I will make like conclusion in Thai. Okay, may I? I won't like make like any mistake. I will try. ค่ะก็จบไปแล้วนะคะสำหรับการสัมมนาพิเศษนะคะจากดร. Felix Moronta นะคะเกี่ยวข้องกับ Biosafety ค่ะก็คือจะเกี่ยวข้องกับการทำrisk assessment แล้วก็ risk management นะคะการประเมินความเสี่ยงแล้วก็การจัดการความเสี่ยงการดูระดับของความเสี่ยงนะในการที่จะเกี่ยวข้องกับพืชที่มีการผ่านกระบวนการทาง modern biotechnology นะคะและหลังจากนั้นก็จะเกี่ยวข้องกับการที่สร้างนโยบายหรือว่า policy ต่างๆนะคะอันนี้ก็คือเป็นสิ่งที่ดร. Felix เนี่ยมาให้ความรู้กับเรานะคะแล้วก็หลังจากนั้นดร. Felix ก็บอกว่าที่องค์กร organization ของ ดร. Felix ก็คือให้คำปรึกษานะคะเกี่ยวข้องกับ Biosafety ได้ซึ่งก็หวังว่าทางมหาลัยเกษตรเนาะบางทีอาจจะได้รับคำปรึกษาในโอกาสต่อๆไปนะคะค่ะ